Welcome back, cell biologists. So we are currently talking about microtubules. In our last lecture, we talked about how the heterodimers can be assembled and disassembled depending on the critical concentration and the actual concentration in the environment. So tubulin is going to bind to GTP. Each heterodimer of tubulin will bind to two GTP molecules. The alpha will bind to one and the beta will bind to one. And the GTP that's bound to the beta subunit is going to have to be hydrolyzed to GDP. That extra phosphate is going to be released after the heterodimer is added to the microtubule. And the GTP is needed to promote the heterodimer interactions and the addition of the heterodimer to the microtubules, but it does not have to actually be hydrolyzed for the microtubules to assemble. If the GTP-tublin concentration is high, it will be added to the microtubule quickly and create a large GTP-tublin cap. And if the concentration starts falling, then the rate of the tubulin addition decreases. So when there's a sufficiently low GTP-tublin concentration, the rate of GTP hydrolysis will exceed the rate of this subunit addition, so the cap will start to shrink. So it affects the stability of that tubulin. Microtubules originate from microtubule organizing centers inside the cell. So microtubule organizing centers are a structure within the cell from where the microtubules commonly originate. The microtubule originating centers are where microtubules are assembled, and they also help anchor one end of the microtubule. During interphase, many interphase cells have the MTOC called a centrosome near the nucleus. And in animal cells, the centrosome will be associated with two centrioles, and around that centriole is a pericentriolar material. We've already discussed the process of mitosis and cell division, and hopefully you're able to remember that. And if not, please go back to your textbook and refresh that material. Centriole walls are formed by 9 pairs of triplet microtubules. So 9 pairs of triplet microtubules. And they attach at right angles to each other. And they're involved in the basal body formation for cilia and flagella. Animal cells that don't have centrioles will have really poorly organized mitotic spindles, but they'll still be able to divide. So they are not critical to cell division, although they are important to helping organize those spindles during mitosis. Centrosomes have large ring-shaped protein complexes which contain gamma-tubulin, and gamma-tubulin is only found in the centrosomes. The gamma-tubulin ring complexes, or the gamma Turks, will nucleate the assembly of new microtubules away from the centrosome. And if you lose those gamma Turks, the cell will be prevented from nucleating their microtubules. There are minus end binding proteins for the microtubules. These are MTOC proteins that function as an anchor. So they don't promote polymerization of the microtubule, they help anchor it in place. One example would be this protein called ninian. Branch microtubules have also been discovered recently, and these form branches that branch off from existing microtubules by recruiting some gamma Turks. And the protein augment helps anchor this structure. So MTOCs nucleate and help anchor the microtubules. And the microtubules will grow outward from the microtubule organizing center with a fixed polarity. The minus ends will be anchored in the MTOC, and the positive ends, the plus ends, will be growing outward. So because of this polarity, there is dynamic growth and shrinkage of the microtubule that occurs at the plus end near the cell periphery. And not all cells have a centrally located microtubule organizing center, and some of them have more than one MTOC. Each MTOC there we go, has a limited number of nucleation and anchorage sites. So there's only so many spaces where they can anchor, and there's only so many spaces where they can nucleate and help create new microtubules. Microtubule nucleating activity can be modulated during mitosis and other similar processes. And centrioles associated with spindle poles in mitotic cells have the highest microtubule nucleating activity while the spindle is still forming. Microtubule stability has to be really tightly regulated by cells, and they use several different microtubule binding proteins to do this. This allows them to very precisely regulate their microtubules. Some microtubule binding proteins use ATP, and the ATP is used to move vesicles, to move organelles, to allow microtubules to slide against or between each other. Other microtubule binding proteins help regulate the structure of the microtubules. MAPs are microtubule associated proteins. These are proteins that can help stabilize, bundle, or destabilize microtubules. They bind at regular intervals along the microtubule wall, which allow the microtubules to interact with other structures. You can see it also comes off here and has an area where it can interact with other proteins. One MAP is called tau. Tau is a microtubule associated protein that helps the microtubules form tight bundles and axons. MAP2 is another MAP that helps promote looser bundles in the dendrites. MAPs like tau and MAP2 have two different regions of them, one that binds to the microtubule wall and one that extends at a right angle. This allows these MAPs to interact with the microtubule and to interact with other protein. And the length of the extended arm will control the spacing of the microtubules, whether they are more closely or diffusely bundled together. There are plus tip proteins, or proteins that bind to the plus end of the tubulin interacting protein. So microtubules can be stabilized by these proteins. They capture and protect the growing plus ends. So some examples of microtubule capture by plus tips at the plus end would be kinetochores that capture microtubules during mitosis, some that associate with the cell cortex, and some that directly bind and stabilize the microtubule ends. There are also microtubule destabilizing or severing proteins. This would include Stathmin or OP18. These bind to tubulin heterodimers. I will say that right eventually. Stathmin and OP18 bind to tubulin heterodimers, which prevents their polymerization, so we're not building microtubules. Catastrophins, we've talked about them a little bit. They act at the ends of microtubules to help peel off those subunits. And catenin and other related proteins that sever microtubules. Okay, that is the end of our microtubule discussion, although we still have microfilaments and intermediate filaments to discuss. If you have any questions, please let me know. And I'll see you for our next video. Bye.